Hello. One of the important uses of Python is in biological aspects is the ability of the scripts and the software to create software for bioinformatic uses. And one of those software could be the use of bioinformatic tools in order to perform DNA digestion. So this tutorial is just about that process. In order to understand how to create a software like that, we've got to go through the following parts in order to understand it. First, we'll go through the introduction to the scope, then the methods of automating the process of creating DNA digestion in Python, and then we'll go through the procedures of one method and give an example, and finally we'll talk about the applications of restriction digestion in the laboratory. So if you ask yourself what is restriction enzyme, they are actually called nucleases, and they are enzymes that are mainly found in bacterial cells, and they use the restriction digestion in order to protect themselves from foreign DNA like viral DNA. There are two main categories of enzymes, restriction enzymes, in bacterial cells. One of them is called exonucleases, which basically trims or cuts DNA from the ends. And the other one is called endonuclease, which has several types. And what they really do is that they cut the DNA from within the DNA sequence, not from the ends. There are three important types of endonucleases, and type 2 among those three is actually the most frequently used one, because it digests the DNA at a specific site, which is called restriction site, and it has the least requirements possible for digestion, which is an important reason to use these kind of digestive enzymes. So if you want to understand the principle of digestion, it is important to DNA digestion, it is important to consider the following animation. So if we have this DNA sequence, let's say this is the 5' end of the first strand, and this is the 3' end of the first strand, which is the leading strand, and we have the other strand here. So this is the first strand and this is the second strand. So if we consider this as the recognition site of enzymes, so restriction enzymes have recognition sites actually. So this is a recognition site of an enzyme which is called XBAR1. The enzyme finds this location in the gene or in the DNA and tries to cut the DNA at a specific place, which is called the restriction site. So this could be the restriction site of XBAR1. So again, this is the recognition site, and this is the restriction site. So we have recognition and restriction sites of any enzyme, a digestive enzyme that cuts the DNA. Upon digesting the DNA at this location, the DNA splits into two parts. So you'll end up having two fragments of DNA. This would be the length of one fragment, and this would be the length of the other fragment. Similarly, you would end up having two fragments if you cut it in any other places. So you would have, like, let's say if you cut it here, you'd have two other fragments, one here and the other here. So if you cut it with two enzymes, you'd end up having three fragments, and the list goes on. This is true for circular DNA as well. So if you have a circular DNA like this, for example, and the recognition site, let's say this is the restriction recognition site, and the enzyme digests it here, so you'll end up having a linear DNA after digesting it here. So this process here is called linearization, or digesting the DNA with one enzyme. Right, so there are several methods of automating the process of restriction digestion in Python in silico, which could be like creating a function or creating an iterator which could iterate through the DNA, and also you could create a function that works with libraries, or you can call a library that has the digestion function already being installed in it or written in it. Our procedure relies on calling a library that comes with Python, and also it has several functions written in it that we can use to digest the DNA, and we write a function ourselves to perform the digestion. I believe this is the easiest way of understanding the process. So the first thing to call is a library that is called RE, which is regular expression library. So you just write down import RE to import the regular expression library, and then we write down a variable to contain the DNA sequence from the user. You can also create a variable that asks for user input, or create a variable that has a default DNA entry. I use this default DNA entry to make life easier. You can do whatever you'd like to. Then I use this function, which is a built-in function, to make this toggled case DNA uppercase. This is just a trick to use it in order to get away from typing errors. So I actually inserted certain pieces of DNA where there are like recognition sites of enzymes, like this one, TCTAGA. I put it here purposely so that I see where it is in the first place, and if it doesn't cut it, I would know that the function doesn't work. Similarly, I have another one here, TCTAGA, which is another restriction recognition site, and the enzyme should cut here when the function works. I also put another recognition site somewhere else for another enzyme so that I can see if it works fine. So I have to have certain variables being defined, like this one as a recognition site. So as I discussed earlier, a recognition site is a place where the enzyme finds it on the DNA. So this one is here. TCTAGA is a recognition site for XBAR1 enzyme to find on the DNA, so that it cuts it right here. The restriction site could be the exact location where the enzyme cuts. So it is also important to know that the restriction site relies inside the recognition site. So it could be here, let's say, within the recognition site. So you have two variables. One of them is a string variable, which is the recognition site, and the other is an integer variable, which is 0, to start from the 0 character and search for the recognition site along the DNA containing variable. Upon digesting this DNA, or cutting this DNA, we need to have fragments. Like if I cut it here, I need to have a fragment that starts from here and ends here, another fragment that starts here and ends here. So I have to take away this one and have other fragments upon digesting it somewhere else if I don't have digestions. So I would have this as a fragment, this is This is actually another fragment, and this would be another fragment. So this would be the cut DNA. Right, so I'd also need to have another variable that is the enzyme variable to hold the enzymes that could be used to digest the DNA. So I have to define a function in order to perform the digestion properly. In order to do that, we have to declare the initiation of the function. So you write down define, or def, name of your function and the parameters. So you have these parameters here to use them later on. So the seq parameter is the sequence parameter that I'm going to use in order to contain the DNA sequence in it. Also, the ins parameter should use the enzyme that I declared earlier. So a parameter could be also described as a variable that can have the DNA or the enzyme in this case. So as I said earlier, the seq parameter can have the DNA variable. The ins parameter can have the enzyme. Here I used input so that the user inputs the name of an enzyme. So I asked the user to kindly write down the name of an enzyme. So I gave the user an example, like X-bar, let's say. So then I do the same thing as I did for the DNA. I make the enzyme uppercase. Then I need to have an if statement so that I work with it. If the enzyme that the user enters is X-bar1, so if it's equal to X-bar1, I wrote it in uppercase because I made it uppercase in the first place. Then the recognition side could be, or should be actually, TC-TAGA. LF means else if the enzyme is equal R1, the recognition side should be this one, GAATTC. So these are the statements that I'm using here. You can have endless numbers of if statements to initiate the enzymes. Following that one, we need to have fragments, as I mentioned earlier. So a fragment could be created by splitting the DNA sequence. So there is a function, a built-in function in Python, which is .split. You write down the name of the variable followed by .split, and then the parameter that you have, or the variable that you want to cut. So we have the recognition side that we use to digest that variable. So we use this recognition side in the split function to digest the sequence. Then we print out something. We print out the fragment actually, but we need to show that the enzyme that we digested with and tell the user that we have the fragment. So we write down for number of fragments, and the I is to be iterated in enumerate fragment. So we tell the fragment. This enumerate is quite important because it tells you the number of fragments counting from 0. We print out the number of fragments and the fragment themselves because we tell the fragment, and end them by a new line. So this is an iterator. So we use the for loop to loop through each fragment that we create here. Now we need to do something to join the fragments and show it as a map. In order to do that, we need to have a variable, we need to use the map function and string function in order to make the fragment a string. How we do that, and why we do that, if you ask yourself. So the fragments that we've created earlier become list. So the list variables could not be counted easily, or the characters are different as in the string variable. So in string variable you can count each character. However, in list variables, when you count or try to find out the length of the list, you would get only one, which is like you have a single character, which is not correct in this case. So what we do is that we convert it to string variable first, and then when we convert it to string variable, we use map function to join each fragment. Now, what we need to do is that we need to remove the last character. In order to do that, we need to have a slice function. This is a slice function of string, so from here to there. And if you ask yourself why we do that, the map and string conversion of the fragment would create a list of DNA with a number at the end, because so if you have the list, like length of the length here, at the end of the list you would have a number here. The number of characters or fragments that you have would be written just after the variable content, and we don't want that. So if we want to remove that one, you write down -1. This is a good strategy to delete one character at the end of the variable that you have, and you put that variable into this variable, or the contents of the first variable into this variable. It could be confusing, but try to print out each statement one after the other to get around it easily. In this case we have 8 new lines, and if you print this one out, the first one out here, so if you print this one out, you'd end up having number 8 at the end of the string. So you have a DNA sequence followed by number 8, which is something confusing. You would not understand this thing in the first place. So now we need to print out the map that we have. So we'd have like a line like this and something here to tell the user that this is the location of the first cut, something like this, another thing here if you have another cut here, and something like this as well. So this would be your fragments, like the first fragment here, the second fragment here, and the third fragment here if you have 2 locations. And you'd have like something in between, like this one, let's say, to notify you about the location of the cut. You can write anything here. You could write the name of the enzyme, like you could write down X bar 1 here to show that you've cut it with X bar 1. This is particularly important if you want to have a general idea of the map of your DNA if you cut it with a single enzyme. And believe me, it is very helpful to do it, and sometimes. On certain occasions you would need to see how the enzyme cuts the DNA before running a gel electrophoresis in the lab. And having your own software for your own enzymes could be a relief. Then you'd return the stringified regular expression DNA sequence, which is basically this variable here. So you just return that one, that's it, to show the user. Now we return the number of fragments first. We also show and tell the user that you'd have these fragments, and then we'll return the map here, and that's it. So let's have a look at the software that we've written so far and see how it works. So we import the module first, or the library, let's say, to be precise. So we import the library here. we define a variable to contain the DNA sequence. We also have this variable to make it uppercase. We need these variables as well, as I mentioned earlier, and define a function here. This is how you define a function basically. It is important to indent your variables and the contents, let's say the statements within your function, and it is critical, otherwise your function wouldn't work properly. All of the variables, the parameters, and whatever you need to work, like print functions, for loops, and whatever statements do you have, should be indented according to this function here. Otherwise it wouldn't work, as I mentioned earlier. So this is the function. You write down these things. So I have this parameter and this parameter. The seek parameter is DNA, so when I run this function, it would ask me for this content of seek parameter. I already told the software that it contains DNA, but I have to tell it that it really contains the DNA for some reasons. And then it would ask me, please enter your enzyme name, for example XPAR1, and I'll just write down XPAR1, and after that it will start to work on the DNA sequence, and the DNA sequence has been written here, and this is it. After that it will start to work on it. Let's run it. You go to run module here, and that's it. So what happens here is that you see nothing. So you write down digestion, open parentheses. Here it asks you for the seek parameter and the enzyme parameter. So you write down DNA as a sequence input and ends as a variable to ask for the next steps. So you write down DNA, comma, and ends, close parentheses. Now you press enter. Here, this is the content of the ends. It asks you to enter the name of an enzyme so that you digest the DNA. So you write down XPAR1. So it doesn't matter whether you write it in lowercase or uppercase, because I included another function to make it uppercase after all. So when you write down this thing, it will tell me that upon digesting your DNA sequence with XPAR1, the following DNA fragments will be produced. So this is neat. These are the fragments. This is the first fragment. This is the second fragment, the third fragment, and the fourth fragment. So these are the fragments, and this is actually the enumeration function. If you recall that we had enumeration function and the number of fragments, then it would tell me the map of your DNA sequence after this digestion would look like the following. So this is basically the same as this one here, but in a different way. So this is the fragment, this is the location of your digestion, this is the location of the second digestion actually, and this is the location of the third digestion. So I actually had 3. So let's double check that. If I try to find TCTAGE. So if I have a look, let's find this one. 3. So I had 3 places where I put TCTAGA, not 2. one of them in lowercase. That's why I had 3 outputs, or 3 places where I cut the DNA. And that's how you perform DNA digestion actually. So back to the presentation. The final point was the applications of restriction digestion. You can use it to perform restriction fragment length polymorphism, and then convert the data to categorical data and perform statistical analysis for population studies. You can do fingerprinting or design cloning strategies and map genes. And the most important things is to design a pipeline. This is particularly important when you want to have a massive study, large number of genes, and then try to find out the relationship between 2 or more genes from different organisms. And also you can write software that perform alignments and create trees for you. Thank you for your attention.